But it's just wonderful to be here and to celebrate. I said the 70th year, Bengt, because where is he? I don't really know when your birthday is, actually. So that's it. Now, I put a photograph up because when we had dinner the other night, several people said you look like a film star. And so I thought, which film star, Hollywood star, would he look like? And then I thought of 007 and Roger Moore, don't you think? And so 007, here we go. The other thing that reminded me of that dinner was that I mentioned that I've had, of course, over the years, many invitations to come to Sweden. This is one of the most marvelous, of course. I really wanted to come, and I've come. But I was invited a few years ago by the biochemistry students at the University of Uppsala. Now, I told some of you this the other night, but I received a letter, and it said, we would like to invite you as an extinguished professor to give a lecture and so on. So I'm not quite extinguished, but so what I'm going to talk about is what Harry Gray said is the most important reaction on the planet. I never had the courage to say that because there are so many important reactions that many of you are working on, of course, with nucleic acids and so on. But and maybe it is the most important reaction on the planet. Planet Earth is a macroscopic reaction vessel. All these complex reactions of biology, and we've been hearing about many of them, unbelievably complicated and sophisticated, all take place in this globe of ours, right? And it's a very special place compared to, at least in our own solar system, that's Mars in the background there, but it's special because it is able to capture sunlight and use that energy of light to drive all these reactions. So you can't build up against entropy without energy input, and that's what we've got. The process by which that energy is captured and made available to the rest of the biosphere is photosynthesis. And the very first reaction of photosynthesis, the actual first chemical reaction, there are primary reactions involving charge transfer and so on, but the first chemical reaction is the splitting of water. And that then, everything else is then downhill. And when you split water, you get molecular oxygen, form a dioxygen bond, and that is released into the atmosphere. As far as we know, all the oxygen in the atmosphere is derived from that reaction. The hydrogen in the form of reducing equivalents is then made available, high energy electrons actually, to drive the reduction of carbon dioxide to organic molecules of life. And also, of course, is the origin of our fossil fuels. This process of water splitting appeared on our planet about 2.5 billion years ago. There's some arguments about exactly when. And I rightly or wrongly, I call this the Big Bang of evolution. For the first time, living organisms had a ready supply of protons or electrons. And of course, we got oceans of water, and this was the major step that has given rise to the variety and the extent of biological activity on this planet. And of course, organisms have from prokaryotic to eukaryotic to multicellular, and then to the complex animals that we know today, and aquatic animals as well. Now, during that process, over these billions of years, oxygen has been evolved into the atmosphere, as I said, and the atmosphere changed from being anaerobic to being aerobic. And it was anaerobic because there was high levels of CO2, and organisms did exist, so there were all the magic chemistry of life was there in the anaerobic environment, and energy was obtained from high energy compounds and through fermentation or by oxidation of high energy compounds like organic acids that existed in the aquatic environment. And of course, Fe2+ would have been a very source of electrons for early biology. But anyway, when water was the mechanism of splitting water was invented, then all hell let loose, and the whole atmosphere of the planet changed, and we have explosion in biological activity. And as that went on, the ozone layer built up, and with the ozone layer, UV radiation was filtered out so that life could explore the terrestrial environment, and here we are today. the process of photosynthesis, I'm giving a very schoolboy lecture here, by the way, is really can be split simply into two reactions. The light reactions, where light is captured, and I've already talked about the splitting of water being the very first chemical reaction, and then the subsequent use of the hydrogen of water, or reducing equivalents, to reduce CO2, as I've already said, and that's the dark reactions. I'm only going to talk about the light reactions, and I'm particularly going to concentrate on the enzyme which carries out that magic chemistry of water splitting, and it's called photosystem 2. And you've heard it mentioned several times, but actually there are two photosystems that work together in tandem to drive electrons for the oxidation level of water to the reduction level for CO2 fixation. So there's not really enough energy in one photon of light absorbed by photosystem 2 to reduce carbon dioxide. So a second electron pump, if you like, is used to get more reducing potential on those electrons. This charge separation in photosystem 2 and 1 occurs across a membrane, and you form a negative reduction compound. One such one is plastoquinone. There are others who are primary acceptors, and the oxidizing hole that's left behind in photosystem 2 is located on a chlorophyll molecule we call P680. It was mentioned yesterday, and it's that oxidizing equivalent which is then pulls an electron out of water, and it starts the catalytic cycle for the production of oxygen. And similar thing happens in photosystem 1. Now, both these photosystems are serviced by light antenna systems, as represented crudely here as funnels, where there are pigment molecules capturing light energy and transferring the excitation very rapidly to the reaction center, to either P680 or P700 in the case of photosystem 1. The two photosystems are redox coupled, and we know a lot about the redox coupling. There's an electron transfer chain between them, but nature never wastes energy, and as the electron flows down the thermodynamic gradient, it drives processes which lead to the synthesis of ATP. So the overall products of the light reactions are ATP and a reducing agent, nicotinamide adenine dinucleotide phosphate, and that is then both of those high energy compounds are used to drive the dark reactions. Now, what I wanted to say before that is that over the years, many people, including myself, started to say, okay, we investigated all this through spectroscopy, and I was involved a lot with a laser spectroscopy with George Porter in particular in London, and we looked at energy transfer and trapping. But a stage came, as it always does with an enzyme, that you want to know what the proteins are. And it turned out the photosystem 1 and photosystem 2 could be separated into separate protein complexes. And it also turned out, as we isolated these complexes. Indeed, actually, I should say they can be partitioned in different parts of the membrane, the photosynthetic membrane, which has made it much easier to be able to isolate them relatively or purely. And Bertil Anderson, sitting here in the audience, was one of the people who, and I'm actually myself, who came up with the concept that the two photosystems were well separated into different regions of the membrane. And that was hard. That was a hard concept because you could not do that without having mobile electron transfer agents between the two photosystems. So after all these analyses, and also genes were identified for the photosystems, and particularly for photosystem 2, we were able to, in the late 80s, 90s, draw up cartoons like this one. And you can see photosystem 2 is transmembrane, as is photosystem 1, and that photosystem 2 and photosystem 1 are linked by a cytochrome complex we call cytochrome B6F, and that's the redox link. And there are two diffusible components, plastoquinone, put in a pool of plastoquinone that can migrate over distance, and also, I should use a pointer really, there's also plastocyanin here, which is a water-soluble protein, but that shuttles electrons from the B6F complex to the photosystem 1. So this is the photosynthetic electron transport chain, and then the way in which the components are organized, as Peter Mitchell emphasized, leads to vectorial transfer of protons one side of the membrane to the other, building up an electrochemical potential gradient for protons, which then drive this wonderful rotor John Walker and colleagues have studied in detail from mitochondria, so very similar to it, to turn ADP into ATP. Now, we've got to understand a lot about this. We've done a lot of molecular biology in my own lab. We did a whole range of knockout mutants of some of these small subunits. There's many there. There's as many as 20 or more than 20 different proteins here making up this complex. And at the very heart are two proteins, and they were mentioned actually by Harry Gray, the so-called D1 and D2 protein. They're very homologous proteins that make up the reaction center where the charge separation occurs, and they also bind the manganese cluster where the catalytic site for the water oxidation process. There were a whole number of other proteins, but not many of them are all that interesting in a way. We don't really know what their function are. They're quite not very boring, but the two aren't. That's CP43 and CP47. They have six transmembrane healers, and they bind chlorophyll. So they're an inner antenna transferring energy to the reaction center, which is made up of D1 and D2. And then there's an outer light harvesting system, which transfers energy of other wavelengths. It binds chlorophyll A and chlorophyll B, and that also services. There's somewhere in the region, it varies on growth conditions, of about 200 pigment molecules, chlorophyll molecules, per reaction center. It is possible, and we exploited this, as did Bertil Anderson, also was involved very much, who's here, but as I've said, in breaking this down into its individual proteins to try to build up this picture. So it's possible, it's like an onion. You can sort of, by detergent treatment, you can remove more the peripheral proteins. And in that way, we were able, ultimately, to sort of have this cartoon with the ordering of the proteins, roughly. And most of the proteins are intrinsic membrane proteins, but there are 3 or 4, it depends on organism, extrinsic proteins which bind over the manganese cluster and seem to stabilize it. Oh, well, let me just, I can use this slide. Now, that's all well and good, but this is where we are with many enzymes, and of course the question is, how do they really work? Okay. And so the next step in the story starts about mid-1994, when PS2 was really a black box in terms of structure. We had cartoons, but not real structure. And I decided that I was going to focus the latter part of my research career. I've worked a lot on photosystem 2, all my career really, but to isolate photosystem 2, which we were able to do easily, we knew how to do that, and start carrying out structural studies. In fact, in my own lab, I stopped all work we were doing on other aspects of photosystem 2 to work on structure. And we very soon, were able, very easy experiments, to take the complex, the photosystem 2 complex, and look at it under negative stain in the electron microscope, and we could see that it was a dimeric complex, which was actually controversial at the time, but it was indeed, with this complex and similar complexes which we made, which were had removed some of the proteins, as I've said, we were able to grow 2-dimensional crystals to undertake this wonderful technique that Richard Henderson and Nigel Unwin introduced of electron crystallography. And electron crystallography at that time was excellent, and you could get down to resolutions of about 8 angstroms, is what we did. did, initially with the help of Werner Kulbrandt when we didn't have a microscope, but later ourselves at Imperial College, with the help particularly of Ben Hankheimer and Ed Morris, we were able to analyze these crystals, and we could see helices, and this is the D1, D2 reaction center proteins with their 5 transmembrane helices, arranged rather like the same corresponding proteins in bacteria, the LNM, in this sort of way. So there was a direct structural homology with the bacterial reaction center, for which received the Nobel Prize in the 80s. And this is CP47 actually. And here we're looking at the transmembrane helices as tubes. We're looking down on it here. And because you're at a resolution of about 8 angstroms, we could actually guess positionings of chlorophylls, because the porphyrin head of chlorophyll is about that magnitude. So we could see that the chlorophylls were actually in 2 layers, and we got some information out of this, and we ultimately were able to map a whole number of the subunits, CP47, CP43, the D1, D2, a lot of these smaller subunits, and the helices for the cytochrome we call B559. And that's where we were in 2001. Well, it's good. We were ahead of the world, actually, but it wasn't good enough because we want sidechain information, obviously. And I turned in the lab to attempting to grow 3D crystals. I had attempted for many years to get 3D crystals of this complex from higher plants, but we were able to isolate the same complex from cyanobacteria, which we also subjected to EM, but we used that preparation to yield crystals. And it's from an organism called Synechococcus elongatus, which is a cyanobacterium. And all this was done with higher plants. And those crystals, in the end, yielded a structure, an X-ray structure. Now, it's a very big complex, as you've seen. It's as a dimer, it's as a dimer, it's about 700 kilodaltons. So this is it for a side view that we published in Science in 2004. All the tubes are transmembrane helices, and here are the extrinsic proteins I mentioned before. They're slightly different in cyanobacteria, but one of them, the PSBO protein, is ubiquitous to all oxygenic organisms, and you can see chlorophylls and so on in there. But and that was many questions were answered by this structure. We're able, for example, to define the protein environment of the of all the pigments and other cofactors in there. So it was a major breakthrough, and it was a dream came true for me, actually. But of course, all the attention in the end has to be, I suppose, on the catalytic center, which carries out the water splitting reaction. You can see that we actually assigned over 5,000 amino acids. It's almost 100% assignment of the amino acids, not all, but we did pretty well at 3.5 angstroms resolution. It was refined to. And this was the cluster. Now, the cluster, you can see, is a very unusual. It was a cubic structure with 3 manganese bridged by oxygens and a calcium. And it was a distorted cube, actually, and it was a cubane, I suppose. And the 4th manganese was a link to that cubane by an oxo bridge. So we had a sort of dangling manganese. And all the amino acids that we've got identified here were ligands for this cluster, according to us. And even today, we know that's true, although there has been modifications of this structure now. Now, this structure was unknown. This type of structure was unknown in the biological literature and also in the chemical literature. Nobody had ever suggested that this structure for photosynthesis or anything else. So it was unique. So there was a certain amount of uncertainty about it, which was became a part of some intense discussions and things around the world, and particularly from Berkeley, where the group there had worked on manganese cluster for years using XFs. Anyway. I just thought I'd show you, I'm very concerned about time because I don't want to go over time, but this is just the the reaction center itself, showing the cofactors. Light arrives at P680, and these chlorophylls basically make up P680. Electron is stimulated to reduce pheophytin, that's a chlorophyll without a magnesium, and then passes to a quinone called QA. It's a plastoquinone. You form a semiquinone. Electron then passes to a QB, and after 2 electrons have gone to QB, it becomes protonated and leaves the reaction center site. On this side, the hole that's generated from the charge transfer is used to oxidize a tyrosine. Harry mentioned that, and we showed that there is a histidine 190 in hydrogen bonding distance to this tyrosine. That was very important because that is the histidine that is tuning the redox potential of this tyrosine. And then the oxidized tyrosine extracts electrons out of this manganese cluster. The other side, you'll wonder, some people will be saying, why does it go up one side? And that's a big question, which we can talk about, if you like, at some time, but not now. But there is the other side. The electron goes up this side. The manganese cluster is on this side. You see there's a pseudo-twofold symmetry axis, but it's almost certain that this side acts as a protective branch in the way that Harry was talking about, that if you have a long-lived oxidizing species here, for example, electrons aren't coming through fast enough from water oxidation, then these are very high potential, over a volt oxidation potential. You have an ability to donate electrons on a millisecond time scale to remove that damage, possible damage by the oxidized species. These reactions are very fast, picoseconds or femtoseconds actually. This one here, about 3 femtoseconds. It slows down here to microseconds and then milliseconds. But again, as Harry says, you've very rapidly separated the oxidized species from the reduced species. Okay, that's that. This is the cubane that we suggested on as best we could with the electron density that we had. And we had anonymous X-ray information on manganese and calcium, and we knew there was, in the end, we knew from the density there were 4 manganese and 1 calcium. And as I've already said, it's a very unusual structure, and we were also able to identify the ligands in the region. I've said one of the most big surprises that was this ligand here, glutamate 354, which came from a big large loop that's on CP43. That was totally unexpected. The others were sort of site-directed mutagenesis had identified some of these as possible ligands to the manganese cluster, and they're all glutamates or histidines or whatever. So and aspartate. That assignment of those amino acids as ligands to the manganese cluster stands today. We drew, at the time in the science paper, we drew this figure to try to get, to show the cubane with its 3 manganese and calcium and the external manganese. And there was a certain amount of vacancy here, and we speculated that perhaps the water oxidation reaction occurred here in this region on the outside of the cubane. And we identified, of course, the pathway to P680 via the tyrosine, and we could identify a channel, a polar channel for hydrogen, the other product, protons rather, from the reaction center, or from the cluster. Now, I'm going to show this, and it's very slow. That's the thing. It just gets you, at least the students or so, the feeling for the structure. This was our original structure, and you can see the chlorophylls. There's 35 chlorophylls there. There are 35 transmembrane helices. They're the extrinsic proteins there, and 30 of the chlorophylls are bound to CP43 and CP46, 16 and 14, and the other 6 chlorophylls are bound in to the reaction center. And you'll simply see those are all the chlorophylls. They're light harvesting. This is where the action occurs, the charge separation. And here you see it. There's a carotenoid nearby, which I mentioned earlier on, which is protective probably. acting, it can be oxidized, it can denote electrons, and there's that twofold actors. This is a non-heme ion midway between the two quinones. We were lucky that the QB quinone was thing. I'm going to stop that because of time. Now, 7 years later, a new structure came along, which was significant because it was done in Japan, in Yokohama, and it was a significant refinement of our original structure because it was at 1.9 angstroms. And at 1.9 angstroms, you could actually see the manganese. We couldn't. We did our best, but we could not actually pinpoint the manganese. They could pinpoint the manganese, and they could also much better describe the ligand field around the manganese cluster. And so this was an important work. It was 7 years later, so we should make some advance, I suppose, in that time. The original paper, actually, our paper, has been cited actually over 2,500 times in just over 10 years, which is not bad. In fact, amazing, really. And I'm sure it's not all in photosynthesis. It's a lot to do with the build-up of interest in artificial photosynthesis and people citing this water-splitting enzyme of nature. So basically, the work was done in Japan. Umeya et al. Published in Nature in 2011, and they confirmed and improved the cubane model proposed by ourselves, and also have been refined in some detail over the years by Per Sigburn using DFT calculations. And his model was more or less confirmed by this X-ray structure. There was always a concern about radiation damage because you generate free electrons in the crystal when you're collecting data, and you've got high valency metal ions in there, manganese 4, for example, and that some of these manganese ions may be reduced, and what we obtained wasn't a real representation. But they more recently used femtosecond X-ray crystallography to avoid radiation damage, and they basically got their same cubane structure with a slight change in manganese to manganese distance, but not that much in the grand plan of things. And that was the structure that the Japanese group came up with. You see it has the cubane structure with the calcium, the 3 manganese, and there is the other external manganese we call manganese 4. And significantly, they had another oxygen bond. Red is oxygen. Another oxo bond from this. We only had the one, so they had 2 oxo bonds. We didn't have that. Fair enough, because we couldn't see that detail. And they could also, at that resolution, see water molecules. There are not many in the intrinsic part of the complex. There's a lot on the external, but these are water molecules, as you see, ligated to the calcium and ligated to this manganese outside the cube. Without spending too much time, that was our original model, 2004, and here we were, 2011, a bit different with this extra oxygen bond, which had been predicted by Per Sigburn. And there is a much better description of the amino acids that we originally identified as ligands, when in many cases they're bidentate. Now, structure's okay, but in the end, we want to know, can we understand the mechanism of this magic reaction of OO bond formation through the splitting of water? That is not an easy reaction. You cannot afford to have long-lived species, oxygen species, which are reactive, because all this is taking place in the very delicate environment of a protein. It's not on a platinum surface or something. So it has to be well managed. And nature well manages it with their 1 photon of light giving about 1 volt. Nature more or less follows this minimum energy curve to produce oxygen. So 1, 2, 3, 4 steps. So you have 4, and it does it by accumulating 4 oxidizing equivalents, each one adding another approximate volt in order to do that. And you can see here the minimum energy is 0.8 volts per electron for each step. That accumulation of charge is accomplished by the manganese cluster, not surprisingly. What am I doing on time? I'm really worried about time. 10 minutes. Oh, okay. here you can see, you do these experiments with photosystems by giving flashes of light, a single turnover flash of light. So you give a flash of light and you convert a state, you could say it's the minimal state, the ground state, S0, to a state S1. And that is, you take out an electron to the tyrosine P680, and then you oxidize a manganese from manganese 3 to manganese 4. And then again, another flash of light, second flash now. Well, don't worry about the numbers, it's not that, it's a bit complicated. But another flash of light, you take out another electron to the oxidized P680, and you convert this again into 3 manganese 4, just 1 now manganese 3. Another flash of light, and you get to a state with 4 manganese 4. Now, I've got a query mark there because there was some uncertainty, but some very recent experiments have confirmed that the S3 state is 4 manganese 4 ions, cations. Then there's the next step, which is the final step of the 4th flash to remove an electron from somewhere, from here maybe, to form a manganese 5. And in so doing, you immediately go to S0, more or less, millisecond. And there is no way to trap that, we don't know of any way to trap that intermediate where the actual OO bomb forms. There's no way, at the moment anyway, nobody has managed to do that. And so we'll now have to look at the whole thing in terms of theory, on quantum theory, or as Per Sigman has, using quantum methods, the density function theory. But there was, originally, I was quite keen on this mechanism because it looked chemically understandable in the terms of this acting as a battery, as the manganese 5 you see, drawing electrons out of a deprotonated water molecule, and then having a nucleophilic attack of another deprotonated water, I've got it here as a hydroxyl, on there to form the OO bond. And the distances are about right to do that. That would be a nucleophilic attack under highly electrophilic oxygen for the chemists. Now, Per Sigman, over the years, has looked at that mechanism. He's looked at many different mechanisms and finds this is not energetically a favorable reaction. And he's found that over many years that the actual OO bond formation involves the formation of a radical within the cube that then attacks one of the oxygens of making up the cube. And that's the way that oxygen is formed. And there is a great deal of evidence for that. Now, there's no evidence for this, by the way, through resonance spectroscopy of different types, EPR and NMR, particularly from a group in Germany at the Max Planck in Mulheim, and the young man there, Cox, who's done a lot of the experiments in Lubit's lab, and they published right up to this year, actually, the various results that really support this Sigman mechanism. And this is Sigman's mechanism. There is the radical formed on the S4 state, and as I've already said, it now crosses and reacts with a nearby oxygen. Has to happen very, very quickly, and then the oxygen is displaced and water binds, and you get back to the S0 state, and then you carry on in the cycle. This movie, which is belongs to Per, actually, Per. And here's where the radical is being formed, and here's the oxygen being formed. It's just repeating itself over and over again. So oxygen radical in the cube. And, well, actually, Per suggested this some years ago, but finalized the mechanism more or less around 2009, before the high-resolution structure. And this was he's followed all the way through to do a full mechanism from S1, S2 to S3, from S3 to S4, but round again. So this is what you saw just now, the formation of the intermediate of S0, the binding of water, the re-establishment of the cluster, and some readjustments with electrons leaving, and ultimately ending up again back in the S4 state with the radical. That was going to be my last slide, actually, but it all depended on time. And I had planned to talk a little bit about artificial photosynthesis. Now, I know I can't talk much about it, but what do you think? 5 minutes? 3 minutes? I think it's enough. and Harry pointed this out in his talk. We are in a situation on this planet where we have used photosynthetic energy to drive all our technology and all the fantastic achievements, and allowed a population of 7 billion people to rise, living at a pretty high standard. I don't know how many motor cars there are on our planet. So it's amazing what we've achieved as a species, but we know we have some downsides to it, and the downside seems to be this continuous burning of fossil fuels, which have resulted from these marvelous reactions, because the byproduct is carbon dioxide, and that's building up in the atmosphere. There is a lot of fossil fuel. There's probably enough for centuries and 1,000 years probably, and I'll tell you why if you need me to, if you ask me. But the problem is this CO2 emission, which is rising all the time, and the question is, can we build a technology which will alleviate this problem? Can we do, if you like, artificial photosynthesis? And that would be take water, split it with all this enormous amount of energy is available to us, 100,000 terawatts averaged over the year, and then split it into hydrogen and release oxygen, just like we do in photosynthesis, and use the hydrogen either directly as a fuel or use it as a reducing agent to convert carbon dioxide into liquid or gaseous fuels like methanol or whatever. So in principle, we should be able to do this with man-made catalysts. So there's a lot of effort going in to try to produce potential catalysts to do this reaction. And the basis of the technique, that the way we're going at the moment, is it should be inorganic, more or less. It needn't absolutely be, but it won't. It has to be a material science problem, really, rather than a biomimicking system. And you would have something that is equivalent to the light harvesting reaction center component, absorbs light, and you get charge separation, as you do in a semiconductor in the conduction and valency band separation, and that use the hole in the valency band to drive the splitting of water, just like P680 does, and you use the high energy electrons to carry out a reductive chemistry, for example, reduce protons to hydrogen. And so a water splitting catalyst here to aid that, and a proton reduction catalyst, or even a carbon reducing catalyst to do chemistry at this end. That's the basis. Now, there's a tremendous amount of work going on to produce catalysts to split water, and also to produce catalysts to make hydrogen. This is actually not such a hard end. This is the hard end. And there was one breakthrough. There are others, and I'm unfair just to pick on the work of Dan Nocera, but he did produce a catalyst, an amazing catalyst, that split water, rather like Photosystem 2 does, and made of cobalt oxide, and it turned out to be cubic, which really excited me. I know Dan quite well, and when he told me, I thought, that's fantastic. This cubic structure was able to carry out, and also this was self-assembling, just like the manganese cluster. To assemble the manganese cluster, you turn manganese 2 into manganese 3, and then somehow magically it self-assemblies, and so does this thing. So it's extremely similar to Photosystem 2, actually. It has many properties which are the same. It can be, it has a low overpotential and so on and so forth. So it's earth-abundant metal, really, self-assembling, as I've already just said, and it can repair itself, actually. We do have a repair system in Photosystem 2, which I haven't talked about. And that catalyst has been, oh dear, that's not the right slide, doesn't matter, has been incorporated on the surface of a three-junction amorphous silicon cell, it's a solar cell, where it's going to use the holes generated from light, and there is a suitable catalyst. It's actually an alloy to make hydrogen. So this is in a sense, an artificial leaf, and is the beginning, a prototype of the type of system that we're trying to work to do not in the lab, but on a much larger scale. In fact, it would have to be on an enormous scale. And this is just that cobalt catalyst, self-assembling cobalt catalyst, splitting water and producing oxygen. I think I should stop there. Thank you very much.
Thank you, Professor Barber. And I think we can allow for one short question. And of course, Professor Barber will also be available during the panel discussion.
Oh.
So the young man in the front.
I was determined to have more questions than one.
Thanks for a fantastic presentation. How did it begin? How did it begin? You have described an enormously complex machinery, but I'm sure nature has, by Darwin's help, evolved it from something more primitive. So could you envisage?
How did it evolve, this
Yes.
cluster?
Yeah.
Is that what you're saying?
No, what was the whole system?
The whole system, how could it be, with all those proteins and so on, how could it evolve? Well, you know...
Not a short answer.
That's not a short question. No,
if I answer the question correctly, can I be a professor at Chalmers? It's like one of those hard questions you ask the students when you're interviewing the other graduates. So, but I can't answer that.
But the Cuban cluster was there from the beginning. You are positive about that?
Well, as far as I know, and I think I bet my life on it, which isn't worth much at the moment, but especially standing watching you, is that this is unique, that there is no variation of theme. Now, you're all used to enzymes which do a certain reaction, but there may be several types of enzymes that do that. Hydrogenases, for example, a whole number of them will do the same reaction. They have iron-iron and iron-nickel hydrogenases. But this is, I would say, if I pass my exam, is unique and is no variation. And the other thing I can tell you, that site-directed mutagenesis of many of those amino acids, which look to be critical as ligands, you can do that. So even remove charged amino acids for non-charged amino acids and still have the magic chemistry of oxygen production. It's extremely robust. And that cube has been synthesized now, both in Caltech by Theo, and also by George Christo in Florida. So and it's very stable. And actually, all calculations, Per Sigmund's up there, and he is one who's calculated that it is a very stable structure. It is ligands, but they're not very fussy about the ligands, actually, although the amino acids are highly conserved.
I think this could also be a part of the panel discussion, how everything emerged on Earth. Yes. So let's thank Professor Barber again.